Okay. So we are happy to have Alessandro Vitriolo, the second speaker from Giuseppe Testa's lab, to speak in this series. Today, focusing on human-specific genes, which of course there are quite few of, which is sort of surprising and not surprising in different ways. But sort of combinatorial disruption of these human-specific genes in using organoids as a readout. So thanks for doing an evening presentation on European time, and we're really excited to hear what you have to say. So please take it away. I can stop sharing if you want to start.
Thank you, Carlo. It's really a pleasure to be here presenting this work, which let me just, of course, there is some issue with the sharing. Can you see my screen? No, you
Yeah,
cannot.
we got it now.
You can? Okay,
Yeah,
good.
your slides are up.
Okay, so I think you're also seeing the slides.
Yep.
Great. So this work has just been pre-printed, and we are hopefully gonna go through revision soon. It's a work that has been in the work for really a few years now, and I'm not going straight into it because I want to give just a brief presentation of also some unpublished data that we have in the lab that Flora has already seen partially a few weeks ago. And yeah, a brief explanation of why we went into this research endeavor in the beginning. So, as you know, so this is more or less the path for tonight. So this evening for me and this afternoon for you. So why focusing on chromatin remodeling genes in general, since it's a key topic of the lab, of Giuseppe's lab. It's then I will give you a brief introduction to the cohort of neurodevelopmental disorder that we have in the lab, and for which we are trying to identify the molecular and cellular convergences, um, generating a lot of data that hopefully, when the work will be published, will be also stored in Nemo for people to play with. I'll give a very brief, very brief example on how NDDs can be informative on recent human evolution and how we try to frame this. And then we will go through first a very short description of how we conceptualized the use of contemporary human uh, models to study evolution across the human conditions. And I will answer the question of how can we interrogate cortical organoids to mask evolutionary relevant genetic interactions. And that's basically the part in which I show the preprint we were talking about. So first thing first, this is a review from the lab from a few years ago, in which we mapped the chromatin remodels that are in the SFARI list, so genes that have been associated or causative of autism. And it's striking because one of the first and most strong enrichments of the SFARI genes in terms of GO is somehow, besides synaptic activity and neuronal functions, it's chromatin regulation. And it's not surprising because, as you can see, all these genes, if mutated during, I don't know, very early in development, let's say in germinal lines or very early uh, in gestation, they will cause a neurodevelopmental disorder. And in fact, these genes are highly expressed. I hope you can see the pointer in the frontal cortex and highly expressed in gestation. So this, of course, makes it easy to imagine why, if you mutate one of these genes, you have neurodevelopmental disorders. So we built a cohort of neurodevelopmental disorders in the period more or less in which I was doing my PhD. And we selected this disorder because they represent a decent spectrum of clinical features, starting from intellectual disability, craniofacial dysmorphism, and also systemic impairments like cardiovascular defects. And on top of that, we also managed to combine disorders that had ASD traits and disorders that didn't show autism spectrum disorder phenotypes. And since our lab is a disease modeling lab, we took somatic cells, mainly skin fibroblasts, we reprogrammed them to iPS, and then we differentiated them into disease-relevant models, which could be brain organoids, let's say iPS-derived neurons of different types, neural crest, and mesenchymal stem cells, because they are able to model early craniofacial development. So, of course, part of this work has been published, including the first description of Gabriele de Vries syndrome, and some of it is in preprint or in preprint very soon. So in general, what I've been doing in the lab in the last years is been to ask these questions. So can we trace a shared molecular mechanism among neurodevelopmental disorders by pinpointing, I don't know, their differential expression, differentiation bias, or cell type specific gene regulatory networks. So in a way, going from NDD genes to their transcriptomes defect, to their endophenotypes, and then trying to correlate this with or causally link them to neurodevelopmental phenotypes. So the first part of this talk today, I will show you a massive experimental work that was conducted by several people in the lab. Almost all the lab was involved, but there is, of course, some key individuals that I need to mention. We differentiated 52 IPS lines independently in parallel into cortical organoids. The patient lines were differentiated up to day 50, while some control were differentiated until day 100. So we have a pretty large description of early neurodevelopment, collecting in total 1 million cells between IPS and cortical brain organoids, multiplexing at least 3 individuals per condition, and the conditions were more or less 10. We found, of course, major macro population. We found IPS, of course, from the IPS. And in the organoids, usually we tend to remove cells that are a bit of targets or strangely differentiated. They account for 5 to 10% of the sequence cells in the organoids. But since we have so many cells, they were enough to make clusters. So we also characterize this a bit, but I won't talk about it today. So we are in the condition to identify individual specific and condition specific phenotypes. And that's basically the key question of this project that is still ongoing. But for now, I can tell you that we are able, through single cell RNA-seq and a bit of machine learning, to identify early and late progenitors and two different streams of neural differentiation that are more or less can be described as lower layer and upper layer neurons. This work, of course, is massive and it requires a lot of integrative analytical work, which, at least for this part, is being conducted by one of our master students, Filippo Prazzoli, who has been mainly involved in this scheme of activities, which of course means to do cell type specific differential expression analysis in each condition, in each disorder, cell type specific gene regulatory network reconstruction to identify with these two master regulators or transcription factors that are able to somehow explain the differential expression that we see in the patient lines. And for the virtue of single cell RNA-seq, we are able, since we have several cell types or cell states, we're able to do disease-specific differential abundance. And on top of that, through machine learning, we're able to do in silico perturbations, so we can somehow answer, at least preliminarily, if the master regulators or the disease genes that we selected, if we can perturb them in silico and kind of inside control lines, of course, and let's say, reconstruct or reproduce the differential abundance that we see in the patient lines. So basically, are the disease genes or the master regulators explaining the differential abundance, at least in silico? And of course, this work is massive because it requires, you can imagine that for each disorder, you can have a differential expression and the gene regulatory network for each disorder and for each cell type. And for each of them, you need to integrate this stuff. And since we have tens of cell states and tens of conditions, this means to do
Can I ask you a question? So,
Yes.
did you integrate all the disorder in a single analysis? And if
Yes.
not, how do you do the differential expression in each disorder? I mean, and also if you do one disorder, so basically, did you consider one disorder separately from the other, or did you combine them?
Good, very good question. So this was one of the open questions that I keep for the summary. But yeah, in this case, in this scenario, what we did was to integrate all the conditions all together. So we have a single dimensionality reduction for all the conditions, including the controls. And every time we compare each disorder separately, each cell type against the control lines of the same cell type, for instance. Basically. So in the end, to make sense of all this work, we take the minimal networks that we can build, which means to take the NDD genes, the master regulators uh, that are connected by one or two steps in the network. So all the NDDs genes that regulate some master regulators, all the master regulators that control NDD genes. Um, so Uh, as I said, what we do is differential abundance as a sort of analysis to reconstruct to which extent we are able to identify a cellular phenotype that is informative of the disorders, at least in terms of differentiation biases or, let's say, um, cell type specific defects. And since, of course, we have several conditions all together in the same place, but we cannot be sure that we sequence the same number of cells per individuals. And also, in the case of controls, we have more individuals per condition. We did some downsampling to make sure that we are able to really compare the right amount of cells of the controls with the right amount of cells of the disorder. And doing so, we have some internal controls because we published already some of this work, and this work is also being mirrored by other groups. So, for instance, in the case of ADMP, that is one of the disease genes that cause as mortal Valderrama syndrome, we have a preprint in which we show also on different, slightly different differentiation protocol that there is an increase in lower layer neurons, and there is published data in mice that show that upon depletion of ADMP, there is a decrease of lower layer and, sorry, an increase of lower layer and decrease of upper layer, which is basically what we see here. So we did this, of course, for the disorder, and we have cases in which we replicated published work or work that is gonna be submitted soon, like for the case of Weaver syndrome. So we have some internal controls that made us think that we are able to also make a description of for the other disorders. And just to make a very to conclude this part, this introductory part, we are doing this in silico perturbations of NDD genes, and this is a very complex plot to navigate. But basically, what we are checking cell type by cell type is that the perturbation of a certain disease gene is increasing or decreasing the amount of the cell type with respect to the controls, the untreated, let's say, unperturbed controls. So in general, we see a good coherence, and we are now trying to develop tools to, of course, integrate this in a more easy to read uh, graphical representation. So in summary, we collected an atlas of early corticogenesis in health and disease, and we will be happy to share it in name as soon as the paper is preprinted or published. And we observe known expected differential abundance defects that make us safe considering the other differential abundance that we observe. We have some disorders that show similar trajectories with respect to the impairment, direct impairment in silico of their disease-causing genes. And it's usually those that show also similar trajectories, which is kind of reassuring. And we identified a core group of transcription factor that didn't show this today for the sake of time, and also to allow Carlo to share this publicly by recording, that probably explain the similarities across disorders, and I'm really happy to talk about this after. So there are open questions. Some we discussed with Flora. Some are, let's say, pressing, like isolating ASD-specific dysregulation, taking advantage of the big, good amount of cells for both conditions, and having multiple disorders with this condition within the same data set. And of course, we are looking for other approaches to prioritize other transcription factors, and this will require, of course, a lot of experimental validation. But as I promised, today we talk about how to integrate the observation and the study of neurodevelopmental disorder with the study of human evolution. So I'm starting from a one slide representation of one paper that we published a few years ago, in which we identified a gene that was shown to have a role in the development of craniofacial features in several animals, and it was a gene involved in one of the key disorders we study in the lab, which is Williams syndrome. And we know that the dosage of the region of the 7q11 to 23 Williams region is associated with kind of opposite traits also from the craniofacial perspective. So the deletion of this region causes Williams syndrome, but the duplication causes another syndrome, which is called 7 microduplication of 7q. So it has several names, but the point is we see upon deletion or duplication of this region opposite features in the modern human face, and so we took the opportunity of having really a lot of samples, because we had back then 3 Williams sample, 3 duplication sample, 4 controls, and also an atypical patient that was sparing the deletion of this gene, BAS1B. So the patient had several phenotypes of the Williams syndrome, but not the craniofacial features, which was somehow suggesting that BAS1B was involved in the definition of the craniofacial features in Williams syndrome. So we also did flag tagged the endogenous copy of BAS1B to track the binding of the DNA of BAS1B through ChIP-seq, and we had really 3 dosages of BAS1B for each genetic background, so 11 genetic backgrounds multiplied by 3 dosages. So we were really able to pinpoint the effect of the changes in BAS1B dosage across the human spectrum. And what we identified in terms of genes that were differentially expressed upon BAS1B perturbation, and genes that also changed in terms of histone binding, histone modifications, histone post-translational modification. That basically the top targets of BAS1B were indeed genes that were selected during human evolution. They were selected in domestication of animals in the situation in which the wild counterpart of the domesticated species showed, let's say, bulkier craniofacial features. So we were able really to pinpoint genes that were regulated by BAS1B and important for craniofacial features. And this was really one of the first time in which we or other groups took IPS-derived models to test evolutive, let's say, theory or hypothesis on human evolution in the dish. So this basically invited us or induced us to start working on human evolution and leveraging all the know-how that we have in neurodevelopment and massive amount of samples that of IPS samples that we have in the lab. And it is important and let's say punctual to use this plethora of samples, because we know that the features that are changing in patients in neurodevelopmental disorders are features that have been evolving through a very large amount of time, with several intermediate phenotypes in extinct humans. So, for instance, we know that the craniofacial features that we consider modern today appeared in the first, let's say, appeared around 100,000 years ago. So, if you take fossils of Homo hominins from different species, you can see that the first sapiens fossils show these features already 400,000 years ago, while the, let's say, the globular shape of the skull of modern humans appeared around 200,000 years ago. And we also see intermediate conditions in which individuals, extinct hominins, or let's say fossils more in general, show either modern faces with, let's say, archaic brains, so elongated skulls, but also we see archaic faces combined with globular shaped skulls. And this means that basically we cannot really so the evolution of these two traits have proceeded kind of in parallel until they stabilized in the form that we see today. And this is also partially justified by the fact that we know now that there was a lot of gene flows across species before the settling of the Homo sapiens species in around 20,000 years ago. So there is a gene pool that is telling us a story of introgression and mixture between different species that we can consider to study the development and the evolution of these traits, these two specific traits these two traits are indeed changing across neurodevelopmental disorders, so it's possible to leverage these patient lines to better study the molecular basis of these phenomena. So the issue with studying modern humans' evolution, one of the issues is that people tend to focus on fixed variants. So variants that are associated to a gene and fixed in all present-day humans. So this means that basically all the individuals in the human population, at least those that have been sequenced, have the same variant in a specific gene, and this variant is not present in extinct humans. So it's a very strong filter that doesn't take into account a number of aspects that we are instead considering. First, it's the unbalanced sample size. So we really have a lot of genomes, millions of genomes sequenced today, but we have only few, 3, 5 high quality ancestral genomes. So we could really risk considering some very extremely rare variants that are not normally captured by the very few ancestral genomes that could still be present. Or, for instance, we don't consider the present day diversity. So the fact that now we are 7 billion people, we used to be much less not so long ago. And another important element is the fact that the genetic variability that we consider today is not the same that was there when we were selected, so when we evolved. So, and finally, the introgression, what I just described in the previous slide. So the fact that there was gene flows between different species. So to take into account all these elements, we focus on nearly fixed variants. And this was an intuition that came from Cedric Book, one of our constitutive collaborators on human evolution. And so basically nearly fixed variant means variants that are found in at least 90% of the present-day human genomes and absent in the high-quality ancestral genomes that are available. So the first dichotomy that appears when you start looking at these variants is, of course, that some of these variants appear at exons, so coding regions, and some other appears at distal non-coding regions, so for instance at enhancers. So we really try to roam these two domains in parallel, and this is part of stuff that I'm going to show you and stuff that's going to be published soon, hopefully. So For instance, we have this work a few years ago in which we try to map through machine learning also the timing of appearance of this variant, for instance, across the last 500,000 years, and to map specifically those variants that might have an effect in brain-specific regions. And so we already have a preliminary map of when and where these mutations appeared and with which effect. And it's interesting to notice that a lot of regulatory variants are in brain-specific regions. So another person that I want to mention is that working in our group is Olivier Leonardi, who's helping us a lot, me in particular, on, let's say, delving into non-coding variants. So when we look at variants that are harbored at distal non-coding regions, it's hard to associate them to genes because of sheer distance, for instance. So, and it's really hard to interpret the effect of a single variant since it's distal, likely on differential affinity that it's generating in transcription factor binding. So we developed somehow a pipeline to do this that has been used already in a paper that has been published and a paper that is preprinted and will be hopefully published soon. And another work in which we are collaborating with the Phantom 6 consortium to identify specifically a subset of this variant that are also impairing RNA-DNA interaction. So we recently published this review in which we discuss how to use different human conditions to infer the role of different proteins or different variants in the acquisition of specific traits. And the first thing that we discuss is the fact that, of course, we can leverage large cohorts of individuals, not necessarily patients, and characterize their differentiation and related to specific variants in specific transcription factors or in their binding sites. And another element that is let's say another strategic point of view that we think is necessary to consider when you work with human modern data and the variability of the human condition to understand human evolution is to identify functional gene modules, so groups of genes that have been associated to the different aspects of human evolution that can be characterized with human models, iPS-derived models, like, for instance, the brain-face decoupling. We know that there are genes that are important for face development specifically. We can look for them and treat them in a specific way, while genes that are involved with globularity, so we can leverage slightly different phenotypes or the variability of a phenotype like the brain case structure to identify genes that have a specific function or a specific influence on globularity, or genes that are expressed or active preferentially in the cerebellum, since we know that the growth of the cortex and the cerebellum is tightly linked, and there is an influence of the development of the cortex on cerebellum and vice versa, which could and likely shape the globularity of the brain. So it is important to consider the expression timing and the location of expression of genes and identify functional groups that could somehow ease the selection of groups of genes to target, because again, we cannot target 100,000 variants at a single time. So we need to find a way to reduce these numbers and cogently treat only groups of genes or variants that make sense. So this is more or less one of our solutions, which is to integrate computationally and experimentally platforms like organoid modeling and multi-layer networks in which we can relate different gene regulatory networks for different conditions, different cell types, with gene to phenotype association derived from literature or from large cohorts of patients, and integrating these two information with the variant the ancestral variants, we can go from the ancestral variants to phenotype by leveraging the connections, the mechanistic connections generated by gene regulatory networks and gene to phenotypes in NDD. And of course, this is made much easier when you can make mosaic organoids or when you can, let's say, maximize by multiplexing the throughput of cortical brain organoids differentiation. That's why I'm citing this very cool paper from Caporale, Castaldi, and Ricoli that we recently published. So now for the last 10, 15 minutes, I will show the paper that is the result of all this reasoning. It's a tale of 15 genes that bear all high-frequency nearly fixed mutations that are missense. So these are genes that have at least one mutation that changes the coding sequence. So it's likely affecting the protein function. And so we start from this rough depiction of Homo evolution, in which we go from different populations that eventually intersected to the modern evolution, to a more precise ramification of sapiens with respect to extinct species, and we take into consideration gene flows to select these genes. So the genes that we selected, that I'm going to show you in a second, show anyway a very strong fixation rate. So this is important because a typical question that people makes is, but you're using nearly fixed, so you're taking into account also potential false positive, blah, blah, blah. So how do you know that these are really proteins that have a different sequence in modern humans? So in a way, this is the genes that we selected are really within the very highest boundary of this filter. So they are really nearly fixed. So 97.5 of known genomes contain those mutations fixed. So I think we are really focusing on genes that make sense to look at with the sapien-specific mutations. And these genes were ranked also based on their presence in homim as associated to NDDs, neurodevelopmental disorders, or show diverse selection patterns. Some had also non-coding mutations associated, different types of selection events. So some genes that were inside introgression deserts or under positive selection, or showing really an excess of mutation. And these genes appeared also in published papers that you can find here and described or even tested through experimental procedures. So we also evaluated the expression of these genes in the cortex, and we selected them also because they were kind of expressed in different cell types. So they were able to capture different functions eventually. And this is the first time in which I show, let's say, the experimental strategy. So once we have selected the genes, what we decided to do was to leverage this protocol from the Butcher Lab that it's based on the upregulation and downregulation of two genes in the same cells. But the original protocol was doing this in a single cell line using viability as a readout. But we wanted to have transcriptomics as a readout. So this meant that we couldn't perturb thousands of genes, hundreds of genes, but we had to really shrink the numbers. And that's why we selected 15 genes. And we had to find a way to know in each cell which perturbations were present after sequencing with the next Illumina. So this is basically the construct that we introduced by lentiviral infection into our IPS, which contains, of course, these lines were already expressing a DCAS9 that was designed for upregulation, fused with viral domains. I think you know the CRISPR-A protocol, or if you know, you can easily find it. And basically, we have two 10x capture sequence, and we basically express the Cas9 only in the cells that have been infected. So we have the Cas9 for the knockout of that we call downregulation, because of course we cannot control precisely the infection rate, and basically we cannot for sure generate full knockouts of these genes. So we have the Cas9 to downregulate the genes, and the CRISPR machinery to upregulate the gene. And we differentiate, of course, the neurons, the IPS into cortical brain organoids with this very common protocol, and we induce doxycycline from day zero. So basically, we harvest and sequence the IPS before the induction. We harvest and sequence the organoids at the end after 25 days. So a very early developmental stage. And we implemented this way of selecting the guides, which I think is worth a mention because I haven't seen it in papers up to now, which is basically to consider not only the effect of the perturbation with tools like Mixscape in R, that basically measuring if the perturbed cells, so the cells that contain a guide, are indeed transcriptionally different from the non-perturbed ones. But we also consider the degree, the rate of detection of the gene, because we know that single cell is, let's say, zero inflated. We cannot detect the genes in every single cell, but globally we expect if there is an upregulation to find a bit more cells that in which the genes is detected, and if the gene is depleted, we expect it to find it less frequently in the perturbed cells. So basically, we are in considering the detection rate and the transcription effect of the guides to select which guides are most likely to be uh, effective. Once we've selected the guide, we basically prepare the virus again to have all the possible combinations of the different perturbations. So if 15 genes are either up or down regulated together with another of the 15 genes, we expect to have more or less n by n minus 1 combination, which is roughly 240. And that's why we stick to this number, because of course, if you think about a single cell experiment and you want to have at least 30, 40 cells for each perturbation or each combination, in this case, to be able to do differential expression, you need to really to have a lot of cells. Plus, you need to consider that even today, the 10x capture system is very leaky, and you can really detect correctly a minority of the cells. So with this procedure, we use also a kind of brute force way to be able to maximize the homogeneity between the different sequencing run and the different differentiation batches. So we basically took 10 organoids per plate uh, of these mosaic organoids containing different cells with different combinations. We sequenced 7 times these pools of organoids, so we have different sequencing batch for different pools of organoids. And we also sequenced a lot of cells on a fully unperturbed organoid set that we integrated within the same, together with the perturbed organoids. So in the organoids that are indeed infected, there will also be combinations of fully non-perturbed cells. And we make sure, of course, at first that the fully non-perturbed cells were aligning transcriptionally with the cells that were not even infected. And that's something plus the single up or single down regulation and the double perturbations. So all in all, the first striking result was that we found really by trajectory reconstruction a few, not a lot, but some unexpected cell types, like some microglia-like cells and some tissues like neuroepithelium, which is typically not found in this type of organoids because let's say the dual SMAD that we use at the beginning, it's kind of bypassing a full neuroepithelial differentiation, let's say, from IPS. So we decided to characterize even more these cell types to make sure that we are not taking making big mistakes. So we took external data, and these are reference markers that we selected because they were highly expressed in our cells and in the reference data for microglia, endothelial cells, neurons, of course, radial glia, and neuroepithelium. In this case, I want to mention OTX2 in particular because it's usually not highly expressed in our cortical organoids. Now, the first readout that I thought would be worth looking at was can we look at cell type specific markers and see if they are direct targets of the genes that we are perturbing? So this is a measure of enrichment in the sense that it's telling you how much the targets of mTOS1 in this example, which is one of the genes that we selected, is containing a significant portion of the cell type markers of, for instance, in this case, macroglia. And so we did this for most of the targets, depending on the availability, of course, of perturbed cells and the robustness of the analysis. And I wanted to show you kind of an external validation of the mTOS1 case, because it really could be a novel candidate to induce microglia formation in cortical organoids, because it is upregulated in microglia derived from IPS from another lab. Its targets are generally upregulated in microglia, and other cases, for instance, there is another gene which is downregulated clearly in microglia. Its targets are downregulated in microglia too. So at least most of them, not all of them. So it really seemed like we are tracking some biologically relevant phenomena here. And if we look at the gene regulatory networks that we are able to build, thanks to our data and our way of integrating it with external epigenomic data and motif databases, here you have let's say a directed graph in which the three of our candidates show their, let's say, show that which genes they regulate, and sorry, you can see that among the direct targets of mTOS1, for instance, there is a clear regulator of neurogenesis that are downregulated, or they share targets with other genes which are known to exert sort of a similar differentiation bias. So I think a key result of this work was also that we were able to pinpoint the genetic interaction between these genes. So to which extent they shared targets across cell types. And we could really target, we could really track cell type specific interactions, which again, genetic interaction, which means that, I don't know, for instance, KIF18A had several targets shared with KIF15. The targets were all expressed preferentially in a specific cell type, this one here. So we were able really to capture cell type specific gene interactions and several interactions across cell types, which means that basically the targets of these genes were shared across two or more cell types. I'm almost done. So of course, after we do this big work of identifying the targets of each perturbation and the targets of the double perturbation, etc., we also want to know if we are able to speak generally about the genes that we were not able to perturb, because we selected 15 out of 50, 60. So are we really tracking something that is relevant across the board for evolution in general? So we took three lists of genes that are basically genes under positive selection in modern humans, or bearing excessive number of variants, or bearing specifically missense mutations in modern humans. And we can see that some genes like HTT and SPAG5 indeed control large groups of genes under positive selection, or we have other genes like these four that are ACAP8, KIF18, mTOS1 that I mentioned before, NO1, which is studied, that is already been studied in human evolution. They show negative correlation with genes that bear a lot of mutations. So they are most likely to be one of those that during evolution have been selected more strongly and required somehow a lot of changes. So the opposite, for instance, phenomena is observed for HTT and K15 and RB161 and SPAC5, which has positive correlated. So I can say positive correlated because basically I'm distinguishing the times in which the gene was downregulated with the times in which the same gene was upregulated. So we really can pinpoint through this system, thanks to the double and the single perturbation, the effect of increasing or reducing the dosage of these genes. And we are doing it in the same organoids in a very controlled system with a lot of replicates. And so these approaches really allowing us to say confidently what happens when you increase or reduce the dosage of these genes. And strikingly, very few of our targets reached a level of significance that was interesting to define those genes as controlling large sets of genes with missense mutations. This is also telling us that probably we should look beyond missense mutations and consider non-coding variants. And I just leave you with a couple of networks that we included in the paper because I think they are informative in a way and providing new inroads into studying these genes in neurodevelopment. For instance, we find that ADSL is able to inhibit DCX, which is a gene involved in neuron migration and is important for neuronal maturation. And I mean, SOX11 is marker of IP of intermediate progenitors. We see a number of interactions that are cell type specific, and we provide them. And most likely, I mean, it's already in Zenodo, it's just not published yet. Gene regulatory networks that you can roam and navigate to see if your favorite gene is involved in a specific regulatory event. And I want to conclude with this slide in which basically I think we accomplished in a way what we wanted to do, because of course we were not expecting to say something key or phenomenal or transformative for each of the 15 genes that we found, right? So among these 15, we found 4 that really are master regulators across cell types. So they really are important for all the cell types and probably for neurodevelopment in general. And that's why they are in the list of genes that have been under selection. And this is probably something on which we will work more in the future. These genes have genetic interactions across cell types, so involving hundreds of genes. They have high interdependency since they regulate each other. There is a clear hierarchy in which SPAC5 regulates K15 and K15 and RB161 are regulating NOVA1. So I think it's really a trope of material for the field. And I want to close by thanking Adriano Scaros, who did all the experiments in this work, Emanuele Villa and Nicolo Caporale, who are, let's say, with me, the senior scientists who are trying to bridge all the projects in the lab, and Filippo, Lorenzo, and Madsen, and Oliviero, who are the people who did a lot of work, especially in the computational experimental side of all the material that I showed today. And all of you for the attention. Of course, Cedric Book for being an exceptional collaborator.
Thanks so much. And jump in, everybody, with questions. This is something, Ali, that I might have missed at the beginning of your description of your present study. So there are 15 genes, perturbed in pairs, up and down. Was this all done in one cell line, I'm assuming?
Yes,
And
we
this
have
is
a single genetic background, which is called Flying, that is public, and people can also easily
so,
obtain.
acknowledging this sort of high throughput craziness that um, is part of this, or a big part of this project. If you wanted to build in cell line diversity, all of a sudden you're multiplying everything by however many, you'd have to do all the perturbations again, or things like that. But without saying, oh, we should just study that more, what are your thoughts on having so much information in one cell line? And would two cell lines do something, or does that, what do you
So,
think that means if you, because you're sampling lots of genetic variants, but only one background, say, right?
so I think it's we are we're touching upon a very sensitive topic because in a way, we have some projects in which we use really a lot of lines, and and people could tendentially complain that we are confounding individual biases with genetic biases, and we don't have enough controls, and we don't have the isogenic lines. And we have this situation which, in theory, we are really working in an isogenic environment, and in theory, we are really looking only at the effect of perturbing the gene, because in the end, we're not introducing the variants. We're not looking at the effect of the variants. We're just trying to fetch the the function of these genes, because these genes are barely studied. So we really are just scraping the surface here. We're just trying to see what are these genes doing and what is their interaction. Of course, having more genetic backgrounds would be better, but for the sake of this specific question, I don't think that having multiple, I mean, it would have complicated a lot
Yeah, no,
experimental design. And I mean, I swear this is a very expensive uh, project already.
no
This
doubt, no doubt.
I mean, one thing we could have been to reduce a lot number of genes, but that the attrition was not so that would have allowed to do, I don't know, three different genetic backgrounds. I don't think so. Would have been
Gotcha.
more
Yeah, it
expensive.
it
If
it
we did
multiplies
six genes,
everything.
yeah, if we did 6 genes, it would have been only 30 combinations, for instance. But still, you have, I mean, you know that, I mean, and maybe you don't know. When we differentiate these organoids, we use 96 wells, and then we transfer the organoids into 6 or 10 centimeter plates. We really are working with a lot of plates, and people get crazy with easily with the amount of material that they have physically to manage. So, uh, well, I think it was a safe, a sweet spot that we found.
Gotcha. Shiraz, you have your hand up?
Yeah, I do. I think that, this topic of, the degree to which confounding variables are interfering with this analysis, as a statistician, I can certainly appreciate that. And I was curious, Alessandro, when you're looking at like a differential expression analysis, right? Do you first perform like a comparison of methodology to see certain methods maybe recapitulating expected neurodevelopmental disorder associated pathways better than others? And maybe that could be something that could, be part of that conversation when looking at the degree to which your analysis and your results are a result of confounding variables.
So it's a very interesting question because it touches on something that it's really what I like to discuss with my students. Are we using the right method? Are we considering the right controls, false positive, false negatives? Where are they? How can we track them? The issue here is that I was afraid of going through a lot of confirmation bias. So what I thought is we just use the methods that have already been benchmarked. So in this case, we use pseudobulk and the jar because there is at least a couple of papers that show in single cell and in bulk that the ability to track the right degree, the right number, let's say, of uh, of false positive and false negative, and also the significance of the results is coherent with the effect size that they are tracking. So, all in all, we just stick to the, let's say, the guidelines of the field. So we used that jar and we used a pseudo bulk wherever we could. Of course, if you had at least 30 cells, I know it's not a lot of cells. So there is a part in the paper in which we discuss this. And we didn't do the differential expression for the combination, all the 240. We did it only for those for which we had enough cells to do differential expression in the proper way. So, I mean, because of course, what you're suggesting would have been a paper on itself.
Yeah, no,
Yes,
and people have written papers about this actually, but yeah, sorry, one last comment, and maybe it's a longer conversation is, while pseudo bulk method is touted as the method that controls for the lowest false positive rates, it's also shown to have elevated false negative rates. That's why I was asking about the recapitulation signal,
Oh.
but that's a conversation for another time. But
No,
thank
but
you.
you're right. You're right. Indeed, we didn't have a lot of differential expressions, and still, with the number of differential expression tables, the number of gene regulatory networks, we still really struggled to identify stuff that was cell type specific, that was not confounded by the fact that, of course, we really have a lot of cell types and a few targets. So I think I'm quite sure we didn't have a lot of false negatives.
Oh,
so
did you have a question? Oh, go.
I have a question. You said 30 cell is the minimum you choose for testing differential expression. Is that right?
Yeah, roughly.
That's
Yeah,
very low. But I mean,
very low.
uh, are you sure that this is reliable? And also, these 30 cells could be different cell types, right? Because
No, no,
you,
no. Yeah, good point. This is this we discuss in the paper. So, to be able to have at least 30, 40 cells per perturbation type, let's say, per combination, we had to stick to this low number and we pooled cells of different cell types. So we integrated, so we put in the design matrix of the differential expression, so the fact that we're coming from different cell types. Somehow. The issue is that, so basically we didn't know very well what to do. Now I'm trying to do some crazy stuff to recover the exact perturbation in cells for which we don't detect the guides. But besides that, the point is um, there is a paper, I think from Aviv Regev, that says that with CropSeq, 30 cells is a good number. So again, we looked for a trade-off that was making us able to do a certain number of differential expression and was backed by other papers. So basically, we described this in the method. We're very transparent about this, and people, reviewer, and you people can tell us, give us feedbacks on how to do better.
Oh,
A couple of two questions. Question number one, when do you look for differential expression within cell types, comparing controls and knockouts, do you see shared gene programs across the cell types, dysregulated in disease?
Yes. Um, but it doesn't strike me as something unexpected. Can you
So
elaborate?
essentially, there is, I think there was a Nature opinion recently saying that there are the gene programs that dysregulate disease sometimes are seen across different cell types. And I was wondering if in your experiments you saw that.
Yeah, we see it. We actually, so we didn't manage to do exactly what I would like to do. So there is a paper. Uh, oh shit, no, I don't remember. I think it's Pinello's lab, which do dicties in which they also build networks along differentiation and in a more dynamical way. I think it would be best, but I didn't know if we had a robust data to do that. And yes, of course, there are gene programs that are dysregulated across cell types, and they could be informative of why certain perturbations induce certain differentiation processes. But I don't think that this is something we can answer with this data. I think it requires ad hoc experiments. I mean, they could be hints, but not more.
Interesting.
Don't draw conclusions from that.
Okay. And then the second question, very quickly, is do you think it's worth to infer gene regulatory networks from data sets that we generate, or should we try to infer gene regulatory networks from big references and then try to see whether our gene regulatory networks are dysregulated? Because otherwise, I feel that everybody builds gene regulatory networks, but if each network is different, which doesn't make sense because then we cannot really interpret it.
So I've thought about it a lot, and the issue is the issues with gene regulatory networks in general, besides the number of methods that exist to do it, to generate them, is that what are you doing them for? So what is your question? So if I have a phenomena and I need to describe it and then interfere with it, uh, having a gene regulatory network say generated from other dataset can be a good benchmark to see if, I don't know, the control lines are reproducing the developmental path that we are looking at. Okay, are our organoids generating the gene regulatory networks that of I don't know, the differentiation of excitatory neuron? Are we seeing something that is consistent with published data and I don't know, fetal data maybe? Yes. Okay. But once you've done that, and you need to compare gene regulatory networks of different conditions in your own data, I'd rather have internally generated gene regulatory networks because at least I can integrate away all the stuff that is different from our model and the reality. Because in the end, these are models. So they must be attuned to the questions that you're asking. And so they need to generate questions or hypotheses that you can test. If I have a gene regulatory network that has a module that is so important for IP to neurons differentiation that is not present in my data because our organoids are not reproducing that phenomenon, it's useless because I cannot test it either in controls or in the patients. So, of course, we can see stuff in the patient lines that makes no sense with respect to the patient individual. But all in all, what we look for is stuff that is reproduced in the experiments, and then there are other moments in which we test this, other moments in which we validate this with other experiment, with other assays, with other differentiation protocols. I mean, there is a ton of things that we could do depending on.
Thanks a lot. Very interesting opinion.
Thank you.
Something I'd like to invite you to do, Ale, is we have an online public data exploration system called Nemo Analytics. If you can join next month, I think we'll end up doing a session on it. So it's not only a place where it is really powerful to have many of the data sets that we all know from the literature in one place, but for instance, your study not only produced the single cell data, but produced many gene regulatory networks, lists of genes that are differentially expressed when you perturb certain genes or gene combinations. And those gene lists, so within the Nemo Analytics system, we can have tens of data sets, big single cell data sets we're interested in, yours and others. But the other thing we can do is overlay on top of that, instead of just looking at one gene at a time, we can look at gene signatures. So for instance, is one of your gene regulatory networks perturbed in, Motri's study of the NOVA1 gene, where he just looks at one gene, or in the autism-related organoid work that Flora does? Are these same systems being perturbed? And we've really designed that NEMO system to be able to look across, it's sort of two very simple things: getting a lot of data together that we're interested in, and then getting gene signatures together. And those two things together allow you, as simple as it is, to ask very many questions. So this is an invitation to you as well as everyone on the call to maybe play in this sandbox together and see what we can start to see as all these data sets come online.
I would love to be able to take part in it.
Great. So I'll be sending out an email. The emails about the next session will kind of give everybody an idea of what that will entail, and so hopefully we'll get some interaction and see how that works all together. But we have a lot of data in already, so we kind of have a critical mass that it's useful to start going on it.
Thank you.
Any more questions for Alessandro? Thanks so much for talking to us today. I'm really glad you could get us into it. And half of your talk was your own work, but the first half was almost more generally about what's going on in Giuseppe's lab and all of your efforts collectively, which was kind of exciting just to see the scope of what you guys have going on with so many different genetically driven CNS perturbations that are going to be super interesting to see as you guys come out with them. So thanks again.
Yes, thank you. See you.
See more of your work. Ciao.
Bye.